The broadcast is now starting. All attendees are in listen-only mode.
So, hi everyone. Good afternoon from my side, and good morning and good evening for all of you that are connecting through a different time zone. So, I am João Borges. I am the conference president chair at the International Younger Chemists Network, IYCN. And on behalf of IYCN and the Younger Chemists Committee from the American Chemical Society, I am very pleased to welcome you to this symposium on entrepreneurship. So thank you very much for joining us and supporting us in this initiative. In particular, we would like to thank all the speakers for their kind availability to share their insights, as well as for kindly accepting to speak at this event. So before going into the program itself that you are seeing in the screen, so I would like just to briefly introduce both networks. So organizing this webinar. So we have International Younger Chemists Network, and you can check all the activities, projects, mission, vision, all the things that we are doing at this website. So you have here is the IYCN Global. Yes?
Sorry, you're not sharing your screen.
No? Oops.
Thank you.
No, yes. Sorry. So as I was saying, so before starting with the program, so you, if you don't know about both networks, so you can get to know more about IYCN at the IYCNglobal.com. You can become a member just by going to this website. So you go directly to the membership form. It's completely free. And you just have to check for the eligibility requirements that you have at the bottom of the membership form, and then submit it to us, and then we'll come back to you as soon as we receive your application. You can also check our activities through the different social media accounts, so LinkedIn, Facebook, and Twitter, and you can do the same, of course, within the Younger Chemists Committee. You have here the links, as well as the website to check more information, as well as if you want to join the committee, you have also here the link to join. So having said this, so today we'll be talking about entrepreneurship. Here I am showing the program, and we have really a great lineup of speakers that will talk about their activities, their entrepreneurial activities, and will provide some insights and advice on how to translate your research from the lab to the market and raise your own business. So basically, we'll be having 3 talks of 25 minutes each, and after each talk, you'll be having 5 minutes of questions that you can make to each of the speakers. So for that, so you are muted. So please write in the chat your question, identify yourself with your name and the affiliation, and then write your question and direct it to the organizers only, please. These questions should be related with the talk itself, because at the end we'll have a panel discussion with all the speakers in which you can make any questions that you might have about the entrepreneurship, what it takes, what are the challenges, and so on. So going back to the first speaker, so will be Professor Javier García Martínez. He's the co-founder and chief scientist of the technology-based company Reef Technology, which develops pioneering zeolites that work as catalysts for several refineries. So he holds a PhD in chemistry from the University of Alicante in Spain, and he conducted postdoctoral research at MIT. In fact, it was during this stay at the MIT that he came up with this idea and launched the company. Javier is also a professor of inorganic chemistry and director of the Molecular Nanotechnology Laboratory at the University of Alicante, where he leads a team that is working on the synthesis and development of nanostructured materials for producing chemicals and energy. Javier is currently the president-elect of IUPAC. He will be the president for the biennium 2022-2024. He's the president of Young Academy of Spain since 2019, and a member of the global young global leader of World Economic Forum, as well as the fellow of the Royal Society of Chemistry. So today, Javier will talk about his experience while as co-founding the Reef Technology Company with a talk entitled A Brief Guide to the Chemical Entrepreneur. So please, Javier, so you have 25 minutes. And let me just give you permission. Trying to find you here.
Okay. So hello, everybody. I hope you can see my screen. I want to start by saying thank you and actually congratulations to both IYCN and ICC for their leadership and for putting together a fantastic webinar about a topic that I feel very close to, which is chemistry entrepreneurship. Today, I'm going to be talking about three different things. The first is a personal story. This is Logan International Airport. This is a few years ago when I was landing in Boston to join MIT as a postdoc, and where I started my company, Rife Technology. So I will tell you a little bit about my own personal story as an entrepreneur. Then I would like to tell you something a little bit different, maybe more personal, which is about social entrepreneurship, a foundation that I created. I founded here in Spain, and is doing something amazing also. So there are also some lessons from that experience as a social entrepreneur. And finally, just a few reflections, something that I learned during the process, and I think it could be useful for, you know, the people who are joining us and maybe learning something about entrepreneurship. So again, everything started at MIT. I went to MIT because it was the best institute to do nanomaterials. And my only aim at that time was to publish as many papers as possible. So I never thought about creating a company there. But I found an ecosystem. I found so many different initiatives that were supporting young students, mainly very young people, to start their own companies, that for me became natural to think about taking my discoveries, the things that I was doing in the lab, to the market. Again, I didn't go to MIT thinking that I will start a company, but it was really a natural conversation for me and a natural process to commercialize the technology that I discovered there. This is Rife Technology. That was a big dream for me when I was a postdoc, and it took 10 years of my life. So this has been a big investment, many long nights, and a lot of traveling. And basically, the vision for me was to apply nanotechnology to a very mature, very big industry like oil refining. Let me start by telling you that refineries account for 8% of all CO2 emissions, and that's a lot of CO2. So if through catalysis, I could make the refining process more efficient, maybe I could reduce the emissions of CO2. And I have in mind that 10% less CO2 emissions just by making one of the key processes of oil refining more efficient, the catalytic cracking process, when crude oil is transformed into gasoline and diesels. If oil refineries would be using our nanostructured catalyst, we will be saving a huge amount of CO2. It's estimated to be around 1.2 million tons of CO2. So basically, just to make it very simple, hydrocarbons are converted into gasoline and diesel and other products through their cracking in conventional zeolites. But they have a very, very tiny holes. What we do with this nanotechnology process is to make these channels much wider, to create molecular highways, so even the very heavy fractions of crude oil could be transformed into gasoline and diesel without producing coke and other waste products that finally will be burned out and produce CO2. So that was the theory. And then I moved to this lab. You can understand that being at MIT and having the most advanced techniques, the best scientists, all the equipment, moving to Bartlesville, Oklahoma, to this lab. This is exactly how we found the lab when we first entered. It was a big shock. This is how it looks like from the exterior. And you may wonder why I decided to leave MIT and move to this area in Oklahoma. It was because of this person, Marvin Johnson. So Marvin Johnson was the first person to receive the National Medal of Technology in the US. This guy, who looks like a very humble senior person, holds 250 patents in my field. So I contacted him and I told him, look, Marvin, I have this MIT patent and I would like to work with you, to partner with you, to see if there is something real about this catalyst. If I can go from these few grams that I prepared at MIT to make something that investors will like to put some money in, and also that we could commercialize. And it took us a lot of time. You know, working in this condition was not easy, but we spent very little money during those days, of course, unpaid, just because I wanted to test if my discovery was actually useful for the industry. And with his help and the help of other people, we have been able to raise several rounds up to a total of close to $100 million. You can understand how difficult it is to introduce a new catalyst in a refinery, in the main process of a refinery, catalytic cracking. So it has been a very long journey. You cannot imagine how difficult, costly it is to produce thousands of tons of a catalyst that is typically sold at $2 per kilogram. So if you put sugar in your recipe, it's already too expensive. So it was a big challenge because of the scale up, because of the cost, because of the industry. It was a big challenge, but we were able to create a well-functioning lab, to hire an outstanding team. And you see our pilot plan when we were able to produce kilograms of our catalyst. It was built from scratch, and to have a world-class lab when we were able to characterize and improve the catalyst that I discovered at MIT. Only one slide to show you that dream that I have, that by using better catalyst, we could reduce CO2 emissions in a real refinery. It has been actually accomplished. So this is a slide from actual data from one of the biggest refineries in the US, the Motiva refinery in Texas. So as you can see here, we predicted some improvements both in liquid volume, more olefins, less gas, less hydrogen, and mostly less coke, 10% less coke. And the coke is what is burned out to produce CO2. So indeed, this refinery, which is now still using our catalyst, saves a lot of CO2, but by using a better catalyst that is able to transform the hydrocarbons more efficiently into gasoline and diesel. And just to give you an idea of the scale of what it means to use a different catalyst, this particular refinery is making between $20 and $50 million per year just because of a more selective catalytic cracking process. I'm very pleased to tell you that last year we sold our company to W.R. Grace, which is one of the main chemical companies, and now they are doing an absolutely great job at commercializing this technology, not just in the US, but globally. This is a global company, one of the leading companies in the production of catalysts, and I'm so pleased to work with them now to make this technology available to everybody. So that was the story of the company I created that I sold last year. But all of this I did while I was in the US. I felt that I have to do something also for my country. So 7 years ago, I started a foundation here in my own country, also with some partners, with some main Spanish companies, to support young talented people. I have to leave my country and go to MIT to find the best opportunities. And I was not able to do that within my own country. So I wanted to provide young talented people from any country, but the ones that decide to stay in Spain with opportunities. And this is what I did. 7 years ago, I put some money, I convinced big Spanish companies to join me, and every year we select 10 talented young people living and working in Spain, and we give them all kinds of resources, funding, we support projects, but mainly we help them to grow both personally and professionally. As you can imagine, everything changed this year with the coronavirus. And again, this is a little bit of a personal story, but one night I got a phone call from one of these young talented guys who told me, Javier, I think we are going to be running out of ventilators very soon here in Spain. I mean, I guess you heard that in the very early days of the pandemic, Spain was severely hit by the pandemic, and he was absolutely right. Just a few weeks after that phone call, Spain, as many other countries, was running out of ventilators. So in a matter of weeks, these young boys and girls decided to work together to build a ventilator that was approved by our national authorities in a matter of weeks. And now this ventilator is a well-functioning ventilator, which is saving lives in different parts of the world. It has been an amazing achievement. This is from national TV when the first ventilator was supplied to one of our main hospitals in Madrid that in April of this year was suffering from a shortage of ventilators. So that made me feel extremely proud. And now currently, this is from the Minister of Foreign Affairs of Ecuador, when some of our ventilators were delivered to the country, and now 6 countries in Latin America have received the ventilators we are producing here in Spain. And I want to share this because I know most of us are chemists, most likely scientists with discoveries, but I don't want to forget social entrepreneurship. I mean, the kind of satisfaction that you get by supporting others to create their own companies and to support their own projects is really fantastic. But I guess the main message that I want to convey during this short presentation is that right now we have this big dilemma, right? So we as a society are making great discoveries every single day, but still we need more solutions. We are going to be hearing in a minute from other entrepreneurs who are providing other kinds of solutions. But the main challenge we have is that between the discovery and the final application, there is a huge gap. A gap that delayed those solutions getting to the patients, or getting to the climate, or getting to just all of us. And I see the entrepreneur as the person who is accelerating the transfer of the discoveries which are produced every day in the labs to wherever they are needed. And that's why, as I told you at the very beginning, for me, entrepreneurship, chemistry entrepreneurship in particular, is so important, because we all read all these fantastic papers, thousands of papers per day. So it's very difficult even to keep up with all the discoveries, even within your own field. But very few of them actually reach the market. And I hope that in the future, more scientists will decide to cross this bridge, and if not taking the discovery all the way to the final customer, at least narrowing, bridging that gap, but taking some step into the commercialization of their discoveries. So now a few lessons that I learned during the process. Again, these are very personal, but I think they are very useful. So how to become a chemistry entrepreneur in 5 minutes. The first thing I would say is that you need to know yourself. Is this really something that you would like to do? Because sometimes we fantasize about entrepreneurship. We make these kinds of presentations and give the impression that things are very easy, that everything is successful. And no, most of the times we fail. I fail many times. I have made a lot of stupid mistakes. And again, it's very long nights, years of commitment. You ask money to investors. You ask your team to believe in you. So you need first to make sure that this is what you want to do, that you are prepared to do that. You don't need to be the CEO of your company. You don't need to take all the responsibility, but you need to be sure that this is what you want to do. So just to be clear, first thing, think carefully if entrepreneurship is what you want to do. The second thing I want to tell you is to think about this long patch, which is very useful. It's very useful at the very beginning. Yes, you have made a great discovery, but then you need to think about how to take that to the market. So even if you have the strongest material, the best solution for climate change, the smallest nanoparticle, wherever, somebody needs to pay for that. So you need to think very carefully about each one of these key questions, right? Which are going to be your partners, your activity, your resources. It doesn't need to be like I did when I was at MIT, like a business plan that was almost like a thesis, like everything written. It could be a working document, but you need to think carefully about what's really your value proposition, how you are going to segment your customers, and most importantly, what's the cost structure and the revenue stream of your company, how you are going to be making money. So before doing anything, before even building a prototype, my recommendation is discover your customer and talk to them. You don't need to have a final product, but you should talk to people that you think they are your customers to see if they really need what you have, how much they are willing to pay for it. And then as soon as you can, produce the first prototype, the first product that you can test, and allow potential customers to give you feedback. Even if you're not making money with your first customers, you want to have them as a reference, you want to have them for feedback. So identifying your customers, again, even if you're not making money out of them, talking to them, learning from them is the most important thing as you are refining your business model. Again, this is 5 minutes, so I'm going to be very brief. So I want to put these questions before all of you who may be thinking about creating a company. Try to answer these questions. Why are you creating this company? It's because everybody else is doing it. It's to improve your CV. It's because you don't have any other choice. Make sure that you have a solid motivation, because many times you're going to be wondering if this is what I really should be doing. The second most important question, as I said, is to have a very clear business model. You need to know how you're going to be making money. And then what's your vision? You know, how you want to change the market, how you want to make a contribution to the industry. The vision is very important, because when you talk to customers, investors, or your team, having a vision is extremely important. And then you need to have a roadmap, a business plan. Even if you're going to modify the business plan, it needs to be refined. You need to have a solid plan, and here it's called business plan, but you need to have a roadmap. You need to know what kind of milestones you need to hit, you know, what's the next step, and what's the final goal. And finally, and this is probably even more important than the technology, is the team, because the technology may fail, most likely will fail, and where you have to solve it is your team. So make sure that you have an outstanding group of people around you who will help you in being successful. And then, because I insist so much about motivation, let me give you a few reasons of why not to start a company? Why not to become an entrepreneur? The first reason I will give you not to be an entrepreneur is because I love the technology. Many times people who make a great discovery go talk to investors just because they want more resources to take this technology to the next step. But no, that is not what you should be doing if you want to start a company. If you start companies to make money and to give back all that investment to the investors. So make sure that you are not confusing a startup with a research project. Once again, also, this is not to advance knowledge. The investors are not going to be happy if what you have in mind is just to advance your career or to understand your technology better. Startup is not a scientific project, and you may be selling things that you don't fully understand, but again, you need to get and hit the market as soon as possible. It's usually quite unlikely that you're going to be successful if you only have a good product, one thing that you sell, because somebody else can be making even a better one while you are trying to commercialize your product. So it's much better if you have a platform technology like introducing mesopores into catalysts. That was my vision. And even better if you have a vision like reducing CO2 in one of the main chemical industries like oil refining. This is a very common mistake, not for young people, but by professors to improve on my CV, because many senior professors feel that they have already accomplished everything. They have hundreds of papers, they are invited to international conferences, but my competitors, they are starting companies, so why am I not the CEO or the inventor or the founder of a company? But that's a very bad idea, because usually the professor is pushing students to start those companies, but there is not a solid business plan there. So make sure that you don't start companies just to improve your CV. And again, another reason I get very often is to be my own boss. Believe me, if you ask for money to investors, if you need to deliver to customers, you are going to have many bosses. So starting your own business doesn't make you feel that you are your own boss. You are going to have a lot of people that you need to be responsive to. So these are some of the reasons why not to start a startup. And then a little bit of my vision about chemistry entrepreneurship. I actually know this guy. He's a very nice guy, Kevin Rose. And I think this the cover of this magazine is not making his justice. But look what it reads on the cover of this magazine. How this kid made 60 million in 18 months. And he's happy, he's young, he's techie, the headphones. So this is almost like what we have in mind when we think about an entrepreneur. Somebody who is very young, who made as much money as possible in the shortest period of time. I want to change that. I would like to see entrepreneurs who are solving real problems, not creating more apps, but actually bridging the gap that I showed you before between the gap between the discoveries and the needs. And I would like to see more women as entrepreneurs. There is a huge gap here also, and I hope that the female chemists that are watching us will be considering also starting their own companies. I'm also worried about this thing, the tower.
2 minutes, please. Okay.
The ivory tower thing. Many times when we teach students, we let them know, the theory, but we don't provide them with solutions. I wrote this article for Science a few years ago about how to become an academic entrepreneur and how we should rethink education if we want to solve big problems. Again, there was an editorial for Chemical Engineering News on the new chemist, how we should educate the new generation of scientists to solve problems not only in the lab, but also in the marketplace. And that involves reinventing chemistry education. Chemistry education is still too much like this. And if you compare this image with this one, taken 100 years ago, you will see it's not much different. One professor speaking to hundreds of students, like if everybody in their classroom will be the same and we are all different. Actually, being normal is boring. So dare to be different. Don't think that becoming an employee or entrepreneur are the only two options that you have. Now you can experience different career paths during your lifetime. And with that, again, I want to congratulate the Younger Chemists Committee of the American Chemical Society and also the International Younger Chemists Network, because you guys are also entrepreneurs. You are solving real problems. You are also getting funding, responding to your organizations, and within IUPAC, we are going to be doing all we can. At least me personally are going to try to be always available to support these kind of initiatives. With that, I will leave you with some resources that I can make available upon request. So thank you very much for your time. Thank you, Joao.
Thank you very much, Javier, for this great talk and guiding tips on how to be an entrepreneur within the chemical science. So we got some questions to you. So the first one is from Raquel Bessa from Brazil. So I will start reading. So in academia, we are used to having our mindset focused on our discoveries for papers and for basic knowledge. Do you have any advice to open our minds to entrepreneurship?
No, absolutely. I mean, if you think that all the education you're going to get is just by going to the classroom, you are wrong. I mean, these days, you are responsible of your own education because information is now available to everybody. So try to expand your horizons. Don't think that your professors are the only responsible people of your own education. Try to attend to these kind of webinars. There are so many resources. Just to give you an idea, the American Chemical Society and MIT, these are just a few examples, offer many, many resources for free where you can even interact with other potential entrepreneurs and find your team. So these days, claiming that what you are getting in the classroom or the kind of environment you have in your university is not encouraging entrepreneurship. It is true. I just mentioned that during my presentation. But if you feel inclined to start your own company, now there are so many resources. When I started Rife Technology, there was nothing like this. And so just take advantage of everything that is happening.
Thank you very much, Javier. So we have a few more questions. So this one is from Dalia. Can you tell us about the catalyst that you have developed? What material did you use? So why have you used as a catalyst? So she wants to know more about that, about the catalyst that you have developed within the company, please.
No problem. So the catalyst is a zeolite. A zeolite is a aluminosilicate, and that's the catalyst that has been used in refineries for over 60 years now. So these zeolites are great catalysts, but they have very small pores. So the big hydrocarbons cannot enter through these tiny pores, and those big hydrocarbons are not converted, and then they are transformed at the end into CO2. So using surfactants, produce micelles, and with that we incorporate mesoporosity, meaning bigger porosity into the same zeolites that the oil refining industry is using today, but just with these molecular highways. So it's a very simple treatment. Basically, we are taking, no, that's exactly what we do. We take exactly the same catalyst that is used today. We put it into a soap solution, in a surfactant solution with a little bit of base, probably pH 10 or so. And after a while, these big mesopores appear, and then the hydrocarbons can go through these molecular highways and be transformed into the desired products without producing so much coal and at the end CO2.
So thank you, Javier. So and last question, will be how to assess if our idea is what it is needed to successfully set up a company.
Wow. Wow, that's a very tough one. Most of the time you are not successful, I would say. Many companies fail, and even in the successful companies, you have many failures. So I would say the three most important things are, number one, your team, the people who are around you, because if you fail, they will help you to solve those problems, those little fails that you may have. The second most important thing is your investors. Investors are also in a way part of your team. So investors may open doors for you to other industries, to put you in contact with other sources of funding. So investors, some people just go for the money, and that's okay. My recommendation is get good investors that feel part of the team. And the third the third more important is yourself, that you feel that you show leadership, that you know what you are doing, that the team is around you, that you feel even an inspiration. So I wasn't the CEO of my company, Joao, as you said, I was the chief scientist. So my main mission, besides improving the technology, was to inspire the team, the customers, and the investors with a vision and with solid technology. That's extremely important. For the scientist within the team, with the founder, you need to be the inspiration for a tech company. So I would say team, investors, and yourself. For me, those are the three more important recipes for success, but success is not guaranteed.
Thank you very much, Javier, for this great talk, and also for your availability to answer to these questions. So please remain with us until the end. So we'll have the panel discussion. So now we'll be moving on to the next speaker. So the next speaker will be Tammy. So Tammy is the co-founder and the chief scientific officer of Hue Inc. So this company was previously named as Tinctorium Inc. This company was founded in 2018. And what the company is doing is making use of microbial fermentation to synthesize dye molecules to provide sustainable indigo dyeing solutions to the textile and fashion industry. So Tammy graduated in bioengineering from the Stanford University in the US in 2013, and has a PhD in bioengineering as well, but in this case from the University of California, Berkeley. So today, Tammy will be talking about the company, what the company is doing with a talk entitled Fermentation-Based Strategy for Sustainable Indigo Dyeing. Please, Tammy, when you are ready. We cannot hear you, Tammy. Yes,
Can you
now
hear me
yes. Yes, please go ahead.
Yeah, thanks so much for allowing me to give this talk to you guys today about what we're doing at Hue. And I'd like to spend a little bit of time talking about the kind of technology that we're working on and some of the challenges that we're facing, as well as talk about the transition from being in grad school to starting a company. So I want to talk to you today about color. Color is everywhere in our world, from cosmetics to textiles to the food we eat. It brightens our day. It's basically everywhere that we look. At Hue, we're focused on using biotechnology and specifically using microbial fermentation to create sustainable dyes. Our first target is indigo. And indigo is a really special molecule. It's irreplaceable as the primary dye for denim. And that's because of a key feature of indigo that makes it different than other blue dyes. Unlike other dyes, it doesn't actually covalently bond to the fibers. It actually forms these big crystals that wedge itself amongst the fibers. And so denim companies actually intentionally flake and wear away these crystals to reveal this white core of the fiber at the very center. And that's how you get this customized fading that makes jeans really popular. If you were to use any other dye, it would just adhere way too well and you wouldn't get these different shades. It's a huge textile dye market because it's the primary source of color for jeans, and it comes in at about $500 million per year. But what a lot of people don't know is that indigo has a dark side also. There are two main problems with indigo. First, the vast majority of indigo used nowadays is produced by chemical synthesis, and it starts with petroleum products. And there's some really nasty reagents that are used in this chemical synthesis, including cyanide and sodamide and a bunch of other things. And the synthesis takes place under pretty volatile conditions. And the second reason that indigo is so problematic is that indigo is water insoluble. So it also requires a lot of this reducing agent to solubilize the dye and turn it into a dye competent form. This reducing agent is really corrosive and also really toxic. So it's harmful for the dye workers who are using, you're making and using the dye. And it also pollutes the waterways where the effluent flows. So the key behind indigo dyeing is this oxidation process right here. You have this precursor indoxyl, which is the monomer on the left, which is really air sensitive, and it oxidizes spontaneously to indigo, which is the stable crystal. It's really important to control where and when this oxidation happens, because once the indigo is oxidized, it's no longer dye competent and it has to be reduced with the reducing agent to be used as a dye. But we thought, what if we could hold this reduced indoxyl state in a stable way? If we can control this indigo oxidation, maybe we can eliminate the use of the reducing agent. So by using a protecting group, we can stabilize indoxyl and make it more tractable for bioproduction. So this glucosyl group prevents oxidation of indoxyl. And when we're ready to actually use it as a dye, we can basically deprotect indican using a beta-glucosidase to remove the glucosyl group and allow indoxyl to oxidize to indigo. This helps us provide spatial and temporal control over when you make indigo. And this mechanism is actually how the indigo plant makes indigo. So we're using this as a fermentation-based strategy for producing the indigo molecule. We have this biosynthetic pathway in the cell that is producing indican. The indican is secreted into the media. And then to actually make the dye, we add the beta-glucosidase. By producing the indican in E. coli, we're basically bypassing both the chemical synthesis of indigo, as well as the reducing agent that's needed for solubilization. This process actually requires only two heterologous enzymes, this FMO, which is an oxidase that turns indole into indoxyl, and a glucosyl transferase that adds on the glucosyl group. So we know that we knew that an oxidase FMO would work well. There was an iGEM team in my grad school lab a couple of years back that had shown that this worked. Its only missing piece was this glucosyl transferase portion. So where can we find a glucosyl transferase that will act on indoxyl? Because this wasn't an enzyme that was known before we were starting this project. So I mentioned earlier that indigo comes from a plant, and this is what was used as a dyestuff before the chemical synthesis was invented and scaled up. So if you see here, for this in this indigo plant, the leaves are actually not blue in an indigo plant, right? Indigo leaves actually have indican and beta-glucosidases made in different cellular compartments. So when you crush up the leaves, the beta-glucosidase will act on the indican and then form this blue color. So we know that since the plants are making indican, there must be some kind of glucosyl transferase in there. So we just have to find the sequence. So the way we did that was basically grinding up some Polygonum tinctorium indigo leaves, extracting the RNA, and then assembling the transcriptome, because the genome and the transcriptome hadn't been known for any indigo plant before then. And this is a project that was carried out by some intrepid undergrads in the lab. And then in parallel, we basically enriched the UGT enzyme from the Polygonum tinctorium leaves by basically fractionating it using protein chromatography, testing all the fractions, seeing what had activity, and then kind of enriching it that way. Once we had a relatively pure enzyme, we digested it and sent it for mass spec at a facility on campus. And then with these two pieces of information, we were able to find this glucosal transferase sequence. So the next step, now that we have theoretically have all of the enzymes that are needed, is to put this pathway into E. coli and see if we can actually make indican and indigo. So first, we've tested out just basic E. coli with no other added enzymes. And we showed that it's only making indole, there's no indican or indigo production, which is as we expected. If we express this FMO in the cell, we also produce indigo. As you can see in the cell pellet, we produce this cool bright color. And this is because indoxyl is just oxidizing out to indigo, but we're not making any indican. If we express both the FMO and the GT in the cell, then we actually decrease the amount of blue color because all of the indoxyl is being sequestered over to indican. And then we're making a lot of product if we supply this tryptophan precursor. And then if we actually add in the beta glucosidase from an external source, we actually see that we can recreate this blue indigo color in the cell media. So this is really great that we can make indican, but we also want to make sure that the product doesn't get degraded in the cell when we're actually trying to make it, right? So we saw that we tested 5 different native glucosidases that were known to work on aromatic substrates or other substrates that basically look similar to indican, and saw that one of the enzymes, BGLA, had some activity on indican. And we also saw that when we knocked it out, it actually reduced the amount of kind of background hydrolysis that we're seeing. And so if we knock out the cell from the enzyme from the cell, we were able to see that the product was a lot more protected when we were trying to make it. So now that we can make indican, we wanted to see if it could be used as a dye. So we first started testing this dye by just kind of spraying fabric with indican and glucosidase kind of broth, and it shows this deep blue indigo color with each successive dye cycle. And then we also took our dye to a prototype dye range in Texas and showed that our dye product can actually drop into industrial equipment, prototype equipment, but still like real actual dye equipment. And we can produce indigo dyed yarns with the same properties that they were expecting in the industry. These dye mills are really normally very huge, so we're lucky to get access to this relatively small machine to test our dye. It was still quite a lot of work to get enough dye to be able to plug into this machine, but we got it to work, and it turned out really great. And this is kind of sort of highlighting the difference between kind of work in grad school and work in a company. In grad school, it was just kind of me armed with 2 spray bottles, and in an actual company, we wanted to do everything a lot more rigorously and repeatably, and use actual dye equipment to test out the dye. And so our strategy is basically to become a recognizable branded ingredient in denim production. So the way that we're approaching commercialization is by starting with high-end, smaller volume brands where we can help tell the sustainability story and bring awareness to this problem of indigo in denim, and bring awareness to how our product is actually solving this problem. Then we'll push towards mass market as we increase our production capacity to be able to bring this dye to more brands in the denim industry. We want this process to become kind of the gold standard for indigo production and eliminate the need for these really environmentally damaging dye production processes that are the current industry norm. And we're really proud of this team that we've built to work together to bring this product to market. So Michelle and I are the co-founders, and we started the company in spring of 2019. And now we have a team of 8 full-time staff across different aspects of R&D and manufacturing that are basically all working on this dye product. We also have this group of stellar advisors, including my grad school advisor, John Duber, as well as a bunch of industry veterans spanning from both biotech as well as fashion and denim design. Now I want to talk a little bit about my entrepreneurship journey and how I ended up basically co-founding this company. So I went to UC Berkeley to do a PhD in bioengineering, and this joint program between Berkeley and UCSF, and I graduated in early 2019. So a lot of this work that I've been showing earlier, especially the E. coli work, was my dissertation project. And so when I was in grad school, I was in a kind of a really entrepreneurial lab, John Duber's lab, and there was always a lot of chatter among the grad students about who was doing what exciting things in the Bay Area startup scene. And a lot of the grad students actually ended up starting their own companies, either off of their grad work or off of other exciting projects. So this route of kind of starting a company did always seem very intimidating, but potentially attainable. And Berkeley actually has a lot of resources for students who want to spin out a company. So near the end of my PhD, I was starting to think about how we could actually commercialize this product. And that's when I applied to this biotech startup accelerator called IndieBio, that's based in San Francisco. And it kind of brings in cohorts of about like 10-ish companies every twice a year or so. And they're all across biotech, including like therapeutics, synthetic biology like ourselves, and diagnostics and everything. And so I applied with my now co-founder, Michelle, and we were accepted into this program in the spring of 2019. And so this program gave us a bit of money to kind of get off the ground and prove out some early concepts. And it also provided a lot of guidance on how to think about business model and how to think about basically commercialization, how to speak to investors, and things like that. So after we kind of graduated from IndieBio, we ended up actually closing a round of funding at the beginning of this year. And it allowed us to really start hiring a team and tackling some of these big challenges. And so the challenges that we faced as a company were a bit different than what they had been in a grad school lab. On one hand, it's great to be focused and have this one goal of developing out this one product. And by talking to people in the dye industry and in the denim industry, we could get a good sense of what our requirements are and what our constraints are and what kinds of price points to hit, what kinds of equipment we had to like deal with, and what we could change and what we couldn't change. But on the other hand, the mindset of kind of working on this project from a company perspective versus an academic perspective, the mindset is really quite different. Everything we do as a company is in service of kind of bringing this one product to market as well as other products in the future, and kind of building out a pipeline, versus in kind of academic circles, the focus was a lot more on what's new and how to see come up with novel ideas and try out novel things, discover new things. And so since a lot of this proof of concept work was done during my PhD, our main focus as a company was on basically scaling up the process and making sure it was robust at a larger scale. One of the pieces of advice that we received was basically that in process development, boring is really good. We don't want any surprises. We don't want anything particularly complicated, ideally not something particularly complicated or new. We want something that's tried and true and can work 100% or close to 100% as much as possible, even at large scale. So during our journey in IndieBio, we were able to work with facilities like ABPDU which is a kind of scale-up fermentation facility in Emeryville, so very close by, that's associated with Lawrence Berkeley National Lab. And we were able to scale up our process from the lab bench scale to 300 liters. And since then, we've also been able to work with other partners to be able to scale up even further. Another aspect that hadn't been developed out quite as rigorously as kind of the biology and the biochemistry was this downstream processing. And it's kind of an area that can be somewhat underestimated in synthetic biology kind of projects. A lot of synthetic biologists often think about how do we make the initial molecule and how do we get good titers and how do we it's basically all about like improving titers on the kind of upstream end. But the downstream is actually really important because actually being able to turn the fermentation broth into actual usable product can be quite challenging and quite costly and requires quite a bit of work to be able to go up to industry specs and be able to provide something that people accept. One of the things that one of the kind of interesting anecdotes that we came across was that when we were first presenting our dye stuff to a dye mill partner, they're used to their normal indigo dye. And when they opened up our dye, they said, this smells very funky because, of course, it came from E. coli. And so that we learned that is something that we definitely need to work on, making sure that our dye is up to spec in all aspects for our dye mills. And I also want to talk about kind of building a team and building out a lab space. I would say this has kind of been one of the most rewarding parts of the journey, and also one of the biggest challenges from life as a grad student. I actually now have a lot more empathy for my grad school PI since I've been learning that even the smallest aspects of leading and managing a team requires a lot of active thought and care, and making sure that basically just like is a lot harder than it looked like when I was a grad student. And so on our team, since everyone's expertise is so different, everyone is kind of bringing a different perspective of the process. And even though, or rather, because the roles are so different and use such different kinds of skills, it can sometimes be hard for everyone to kind of talk on the same wavelength, but it's been a really rewarding thing to have everyone else. Each member of the team plays such a crucial part, such a crucial role to the success of the company. And it's great to see everyone coming up with new ideas and working together to make this project a reality. This is the kind of difference in background is I found to be a little bit different from being in a grad school lab where a lot of us were kind of had different backgrounds, but did have a lot of the same mindset. So kind of having people across different disciplines was really useful for our team. And also this is a photo here of the lab space that we're building up. It basically started from nothing, and we're really proud to have built up this working lab that we can do experiments in and test out all of the processes that we're thinking of, and really make some great progress on our own. So we're excited to work together to culture nature's rainbow and create a brighter future for the planet. And happy to take any questions at this point. Thanks for listening.
Thank you very much, Tammy, for this great talk on this technology developed by you on using a green chemistry-based approach towards sustainable indigo through genes. So we do have some questions for you. Well, the first one is from Dalia. She's asking what kind of plants produce indigo and what is the known toxicity of indigo dye for humans?
Indigo dye has been used for a long time and has some toxicity at very large scales, but it's not really the indigo dye molecule or the indigo molecule, I guess, that bad for humans. I think people use it all the time, and it's not been shown to be particularly toxic. It's a lot of the side products from the synthesis process and a lot of the ingredients that are used in the chemical synthesis that are carcinogenic and toxic and pretty bad for people. Is that kind of answering the question? Oops.
Yes. So I also have one or two questions. So how far is the process of reaching the market? And curiosity, how many liters of bacterial culture are needed to dye a pair of jeans?
Yeah, so it's for the first part of the question of how far are we in the process of kind of scaling up and pushing out a product. We are kind of in the midst of scaling up the process to be able to launch a first product, which we hope to launch sometime in next year, although with COVID-19, it might be pushed back a little bit, but we're still hopeful that we are going to launch something sometime in maybe the second half of next year. And as it comes to how many liters of dye can dye a pair of jeans, it kind of depends on where our titer is right now. I think right now it doesn't take that many liters. I would say like a couple of liters to dye a pair of jeans. However, since these dye mills are so enormous, there's no way to dye one single pair of jeans. Unfortunately, you have to make huge amounts of dyes to like kind of power a dye mill to produce lots of pairs of jeans at once.
Okay, thank you very much. So the next question is by Juan. So he's asking, how do you develop or get prepared the pitch to the investors?
Yeah, I think it comes with a lot of practice and basically just doing the pitch over and over again and kind of gauging reactions and seeing where people are confused. This is something that Michelle and I worked on a lot, especially through the IndieBio process. Basically, a lot of the time during IndieBio was basically practicing the pitch and kind of honing the story to be something that investors really resonate with, and also, of course, stays true to what you're doing as a company. So yeah, I guess just say a lot of practice.
Thank you. The last question is made by Rodel Cuero. I hope I read it well. So would it be possible to use the same approach to another dye? So to get another color?
Yeah, so there are actually interesting other molecules that we're looking at to expand beyond indigo dye. Although we want to be focused right now to actually like launch a first product, we do have kind of other colors in mind. I would say this process is can be expanded to other colors that are related to indigo, like structurally related to indigo. If you modify the indigo compound, you can actually make other cool tones, other kinds of colors. So we see this being useful for those colors also. And then kind of branching out, we also see the ability to basically glucosylate these small molecules to open up kind of a world of possibilities for other kinds of dyes in textiles and food dyes to make other different kinds of colors.
Mm-hmm. Thank you. So on the final question, what are the main hurdles or challenges behind the fermentation process that might potentially inhibit bacteria from growing?
Yeah, so I think our fermentation process we've been lucky that it has been pretty easy to deal with recently, or I guess so far. I think some of the challenges that we will anticipate and or have heard from other people can be a problem is basically scaling up to larger tanks. Sometimes the mixing will be not as homogeneous as smaller scale, and sometimes will be harder to supply things like oxygen to the whole tank. Even though the process you developed out at the bench scale, it's easy to control and everything kind of behaves as expected. So I'd say scaling up is one tricky part. I think sometimes cells can make rather toxic byproducts that can kill the cell. For some organisms, it can be something like acids in general. Acetic acid or other kinds of organic acids are also something that can be pretty troubling for fermentation processes. Yeah, I think, yeah, basically just improving, for us, it's currently just like trying to improve the titer in any way and removing bottlenecks. That's kind of our main focus.
Thank you very much, Tammy. We do have a few more questions, but due to the sake of time, we'll move on. We can make them later at panel discussion. Thank you for being with us. So now I would like to present the last speaker of this session before the panel discussion. So the next speaker will be Andrew Watson. So Andrew is the founder and the CEO and chairman of Ligando, which was founded in 2014. The company works on regenerative medicine and is specialized in the design and synthesis of biomimetic peptides for gene therapy, cell-specific targeting, vaccines, and antiviral defense. Since 2020, the company starts to work on pandemic defense technology, and the company is developing peptide nanoscaffolds to work as a combination of antidote and vaccine to the SARS-CoV-2 coronavirus. Although the company is also working towards our other outbreaks. Andrew received his undergraduate training in biomedical engineering from Rensselaer Polytechnic Institute, New York, in 2013, and has served as a visiting scientist at the Buck Institute for Research on Aging in 2015. So today, Andrew will elucidate us on the pioneering peptide technology that is being developed at Ligando, with a talk on the rapid simulation, design, and synthesis of SARS-CoV-2 antidote peptide nanoscaffolds for pandemic defense. Please, Andrew, when you are.
Awesome. Thank you for inviting me, Zhao. And forgive me here. I just want to figure out, where is the screen share button?
And now you should be able.
Okay, there we go. Can you see my screen?
Yes.
Awesome. So as mentioned, I'm Andre. I'm a founder of Ligando. Let me just make this work. There we go. So Ligando's a company that was founded in 2013, really 2014. I've been working on this company since I was 21. So originally this work was really founded to develop these technologies that could be used for delivery of genes and gene editing tools to specific cells. And as we progressed, we really sought to build out a platform that could target and manipulate essentially any cell with any receptor. So a number of years went into sort of this technology development for a platform that could do such a thing, because the technologies didn't really exist in a cohesive way. So we had a lot of sort of lipid delivery technologies. Gene editing was just taking off. CRISPR wasn't even really a thing at the time of when I first started the company. So we had like precursors to CRISPR for gene editing, and there was a clear market need, but also it was kind of like looking at, you know, if you were to go back to the 70s and try to explain like personal computing or something, you know, you knew that it was going to happen, but also that a lot of fundamental research had to happen. And I sort of entered this knowing that this could be easily a 20 or 30-year project. So the question was, how do you, to some of the earlier points that were mentioned, you know, you have to balance when you start a company, having a product and focusing on building something that can actually be, you know, sold, that the market needs, with also doing this technology development, which a lot of people do in academia, but there is some balance to be struck there. And if we look historically at what made Silicon Valley great before it became, you know, the land of apps, it was that companies were willing to do that hard research. So if you look at companies like Intel, IBM, even Apple and Microsoft, they were willing to actually do fundamental research into nanotechnology. And to this day, you see these companies every year. Can we make the, transistors a little smaller? Can we make this process a little faster? I think that we've kind of lacked that in biotech because there's such a long development timeline. So really the vision of Ligandel is to be this long-term company that can do this technology development. And that's not without its difficulties when you're fundraising, which we'll talk about later. And I think a lot of the earlier points in the symposium are also really good points about sort of what investors want versus, what you might need to do sometimes to build a hard tech company. So in any case, I'll walk through some of the work that we've been doing most recently, as well as work that preceded that, and really the gene therapy, gene delivery work that we were doing and that we still will do after we develop our first product for COVID here. So most recently, we sought out to develop an antidote to SARS-CoV-2. So on the left here, the figure that's labeled A shows sort of the virus and its spike protein specifically. So if you imagine a virus has, oh, here we go. Imagine a virus, each of these little things is the spikes. And it's that green thing on the left. And it binds to ACE2, which is the receptor that the virus uses for cellular entry. That RBD is the receptor binding domain. And then figure B here in the middle shows that receptor binding domain bound to ACE2. So you're kind of going from something that's 1,200 amino acids to something that's a few hundred amino acids here. And then finally on the right, that panel with 5 figures in it shows these peptides that we built. So we created these short peptides that mimic the virus and how they bind and can compete with the virus for binding to the ACE2 receptor. Furthermore, we published this, and you can read about it in this bioRxiv open access publication. But basically, we discovered that ACE2 prevents antibodies, which are naturally how your body and your immune system survey for viruses, in addition to T cells and the innate immune system. But antibodies critically are able to bind to the virus and actually flag it for destruction. So it turns out that ACE2, this receptor, also exists in soluble form. So it's floating around in the blood and actually coating the virus. And anyway, we showed that neutralizing antibodies cannot bind to the virus in the presence of ACE2, which is problematic because it means that the virus is basically not like other viruses in that it gets cloaked. So it gets coated in this ACE2, and then it's basically invisible to a large part of your immune system. This might have something to do with why the neutralizing antibodies, which are the antibodies that you need to actually neutralize the virus and prevent it from binding to the cell, versus other antibodies that can form against sort of the sides and different parts, but not necessarily serving a function to neutralize the virus. So anyway, the people who are getting infected and recovering from SARS-CoV-2 are having either low or undetectable neutralizing antibody levels in a double-digit percentage of cases in as little as 30-something to 50 days, in up to like 33% of people, depending on what cohorts you look at. So we sought out to develop something that wasn't just, a lot of groups are looking at how to target the virus directly. So, you have ACE2, people using ACE2 bound to like antibody fragment, an FC fragment to bind to the virus and eliminate it. You have like nanobodies derived from llamas, you have antibody therapies, and other approaches for targeting the virus directly, right? But the issue is that those therapies, for the most part, are actually hiding the part of the virus that you want your immune system to recognize and bind to. So the idea here was to develop the complement of that. So rather than mimicking something binding to the virus or building something to bind to the virus, could we mimic the virus itself to occupy the sites that the virus would bind to? And so, to my knowledge, this approach hasn't really been employed, where you're building these peptides, in this case, a synthetic peptide, as a scaffold that inhibits the virus from binding, but also exposes the virus for enhanced immune recognition. So not only do the peptides bind to antibodies, but by taking off that soluble ACE2, the virus should be more visible to your immune system for neutralization. So this is sort of what we developed, and we just published this paper in an open access preprint server that basically, with UCSF, we showed both the binding behavior of these peptides that we made, and also that they can inhibit the ACE2 cloak, that they can bind to neutralizing antibodies, and furthermore, that they can inhibit infection in something called the pseudotyped lentiviral assay. So you take lentivirus, which is like HIV, but modified so that it doesn't insert copies of HIV. Instead, it inserts a fluorescent, or in this case, luminescent gene. And the outside of that virus is coded in the spike protein instead of HIV stuff, which means that then you can infect cells that have ACE2 receptors and measure the infection. So we showed like 95 to 100% inhibition of the infection with these peptides. But in order to get to the point even that we could do this, because keep in mind, this wasn't what we started off with. We started off as, you know, a gene delivery company building targeting ligands. But the critical part is that we were building these peptide targeting ligands. So when I received the sequence of SARS-CoV-2 back in February, I was able to utilize that sequence in the absence of structural data, using something called Swiss model, which is a server for modeling what things should look like based on amino acid sequences. And we had the structure of SARS-1 from 2003 and 2004. So here, this green thing is actually a simulation of what the spike protein looks like that we were able to have before that structural data was available. And I docked it to ACE2 and actually figured out which parts are critical for binding. And this was something that we were doing before for other targeting ligands. So we've really built kind of this broader technology for, you know, here's a cell, here are surface markers, I want to target that. And now I can either target it, or I can also target it and deliver something else, which leads me to the next point. So this is really what we had developed before as sort of this multi-stage peptide-based delivery technology, where we built these sort of multi-stage nanoparticles that could target a cell, survive that initial environment in the digestive compartment of the cell, the endosome, escape from it, and then optionally deliver to the nucleus. And you know, this required a lot of building robots and software to control those robots. And you know, we spent quite a bit on all the equipment, on all of the foundational R&D to be able to essentially input a set of parameters and output, you know, 100 nanoparticle formulations, because we built this self-assembling nanoparticle platform. And actually, Chemical and Engineering News, which is owned by ACS, published this on the bottom here. I'll link to it later, but it was an article about the work we were doing. So in any case, we built out this infrastructure where we could make a nanoparticle, we could make the peptides, we could screen those nanoparticles for their physical properties, we could screen the biological properties of what those nanoparticles do in terms of gene editing or delivering genes, things like cell viability, survival, you know, transfection efficiency, and then basically do those screens. And for a long time, in a way, because bear in mind, you know, this is many years of R&D that normally you would see done in just like an academic environment, or, you know, in a big company and in some sort of like skunkworks lab. And here it's like, well, this is a startup and we're supposed to commercialize these things. It's not a science project. At the same time, we need to do scientific research and discover something new in order to build a product we want, because it's at the bleeding edge. So we actually sought to tailor this to, so backtrack a little bit and sort of talk about my story. When I was at Rensselaer Polytechnic Institute, RPI, in upstate New York, it's the oldest polytechnic university in the United States. I was studying biomedical engineering. So the first year and a half of sort of pre-Lycandol, I was still in my undergrad. So it was like 19, 20, turning 21. And I built this delivery technology based on peptides and kind of a silica shell to deliver talons, which were a precursor to CRISPR. Talons are like a protein-based gene editing tool, which we tailored for this bone fragility disease. And ultimately this culminated in this study, which was in osteogenesis imperfecta mice that have these collagen mutations, so their bones are fragile and brittle. And we showed that injecting the nanoparticles into mice could result in approximately 50% efficient gene edit and thicker bone. But then, you know, it was kind of a question of commercializing this. And I bought a one-way ticket to Silicon Valley when I was 21, sort of just left academia cold. I'm like done. You know, I was considering going to get my PhD in neuroengineering, and I dropped everything. And I sort of came out here August of 2013, and it was like, all right, I'm going to figure everything out. So I'm going to patent this through my university, license it, got to figure out how to raise money. At the time, there was not this sort of booming startup biotech scene in Silicon Valley. I mean, obviously you had your big companies, your Genentechs, all of your, you know, pharma companies, larger entities out here, but there weren't these incubators. Like Y Combinator didn't have its bio program in 2013. Indie Bio didn't exist yet. Like I knew Ryan Bethencourt and Ron Chigetta, the founders of Indie Bio, back when they were like founding Berkeley Biolabs and stuff. So running around the valley being like, you know, hey, I'm gonna build nanotech that's gonna deliver this thing called CRISPR, which you haven't heard of. We're building ligands that target cells, and we're building these nanoparticles that are gonna do stuff. And we wanna tailor it initially to like rare genetic diseases and maybe Huntington's disease. People are just like, you know, you're crazy, right? So maybe it was. Maybe I still am. Maybe that's why we keep doing this as entrepreneurs. But it took a while to, you know, even establish like the first lab. You know, as mentioned, I was a visiting scientist at the Buck Institute for maybe 6 months, and that involved actually working on delivering CRISPR as a plasmid form with nanoparticles and trying to do this in Huntington's patient derived neural stem cells. And then trying to raise money for that was a nightmare. Applying for SBIRs, people are like, this is too advanced, too bleeding edge, you know, no one would give you money to do this, especially, you know, given the state of the field. So then this was where things kind of got interesting, because transition to 2017, you know, this is like already almost 4 years after I came out here, 3 years after starting a C Corp properly. And at the time, something called T-cell immunotherapy had already been, you know, in various stages of clinical development and whatnot for a while. I mean, really since perhaps even the late 90s, the first ideas of engineering these T-cells to have like new receptors on them to target cancer cells, right? And in 2017, these two companies, Kite Pharma and Juno Therapeutics, were sold to Gilead and Celgene for like $12 and $7 billion respectively, which sort of was the first instance of gene editing and gene therapy in an immunotherapy context becoming a viable thing from a market perspective. And this is where, when you're starting a startup, you have to really consider the market readiness of what you're doing, because you can have this advanced technology or these ideas or, you know, a great rationale for what you're going to do, but ultimately the market has to sometimes catch up to what you're doing. And you can go around and talk, and you're basically just educating VCs, and they're not paying you for it. So the thing to consider is that the moment that became something that suddenly there was an exit, there was a comparable deal on the market. And furthermore, the first gene therapies were approved in 2017. You started seeing like Avexis with Novartis and all this other stuff just pushing through. A lot of it being viral vector-based stuff, but it didn't matter. Suddenly, people look at my company and they're like, oh, that makes sense. Yeah, like let's put some money. So I raised $2 million in 2017. Up until that point, it was all like small checks, maybe up to 250K angel round here, there, but it was never more than enough to have like a single bench running around to different labs at UC Berkeley, whatnot, and trying to just like perform experiments in a very piecewise fashion. And then 2017 came along. Can talk a lot about sort of the things that investors tried to do before that point. Like I had survived 2 attempts or more of taking over my company, or people trying to do what we call like hostile takeovers and things like that. So bear in mind, you also end up frequently spending majority of your time on dealing with people who don't necessarily have your best interests at heart, and they see an opportunity to make money. They want to make money in 2 or 3 years. Maybe that's great if your startup is like something where I built this thing, I want to sell it. Great, go do that. But sometimes you want to build something, as mentioned previously, like a platform or something that could be extended to all these other things. And maybe it is something that you feel like if you just sell it off, it has intellectual property, it has all these components, like that's going to be lost and it's going to be put into just a couple of things. So you want to be kind of the steward of this technology where you're willing to take all these punches, these blows, because you know that ultimately you're the only person who's going to have the integrity to build that thing to completion. So you can sell this piece or this piece for this application if you think that will get it to market, but maybe you don't want to sell the whole company, and that's not that controversial. So anyway, we ended up raising $2 million, building out our own lab in San Francisco, investing in over $1 million worth of equipment. And this here was a really cool image taken with super resolution microscopy. The green stuff is CRISPR, and this is a T cell. The blue is its nucleus. And basically, if you can introduce the CRISPR to the T cell, then you can edit it. And there's more to the story. You need to insert a new gene to actually get a T cell to go attack something. But here we showed that we could target like the T cell receptor and delete it. And it's the first step before you insert something back. And we did this with peptides, which no one had done previously with CRISPR. So we built this peptide-based nanomaterial that could self-assemble and form a package in water around this, this CRISPR-Cas9 ribonucleic protein. And ultimately, then you could modularly shift out the targeting molecule to target different cells. So it's not just for T cells, but here we show the T cells, and which achieved like 34% efficient gene editing, which is better, still is better than anything I've seen published with like a non-viral, non-electroporation based technology for editing these cells. And
sorry, Andre, 2 minutes, please.
Oh, great. Yeah. So we've been featured in some articles you can read if you're curious to learn more. We have 45 patents filed now, most of them owned by Ligandel, so it's like a very long-term thing. And just to kind of briefly summarize, starting a company, building a technology in academia versus in a company, fundraising for a company, all this stuff, it has its cycles. So I think nowadays, as mentioned, you have incubators like IndieBio, like YC. They definitely help you to raise your valuation, develop something quickly, try to get a product out there that could be sold, and offer sort of their network. And one of the keys for fundraising is really that you need to create the leverage. So if you go out and say, hello, Mr. Venture Capitalist, I'd like to raise some money for this thing, maybe, but they're going to try to mess with you. If you go out and you say, I'm building this product, just want to talk, any thoughts on this? That's where you want to be, right? Like you actually don't want to be fundraising if you want to fundraise. Kind of funny. And certainly don't get into something if you're not willing to deal with, you know, obviously a startup shouldn't be your passion project, but at the same time, if you're going to spend 3 years, 5 years, 10 years, more than that on a company, it needs to be your baby. You're going to burn out otherwise. Like, there are so many things that will get thrown at you, and they're not the things you expect. When you start a company, you think, you know, it's going to be my product, I got to build it, that will be hard, and then I'll figure it out, and then people will give me money, and then we'll build that product and it'll be great. You know, why wouldn't it be that way? We're trying to change the world or solve this big problem. Why wouldn't everyone want to help? But then you encounter like human greed. People care more about stealing information or gaining access to information or kind of waiting it out because they can. And you have to deal with that. Like, that is very much so what running a company is about, frequently, not just, you know, building things. And you have to persist. You have to stick through for many years. And, you know, there will be times when you wake up in the morning and you're just like, wow, like, how do I do this? Like, I got to keep going. And there are also times where you feel amazing and you're just like on top of the world. And I think taking care of yourself is really important. Like, if you don't practice self-care, you're not going to persist in your company. So if you go and start a company, just, you know, be prepared for what you're signing up for. But it's really a very valuable journey, and it's something that if you want to solve a hard problem, I think it's one of the best ways to do that. So thank you. That's all I got.
Thank you very much, Andrew, for this great talk and for describing your pathway as well as what your company is doing. So we have a couple of questions. So the first one is from Denisa from Slovakia. So she is asking, how did you know that your company is going to be successful? How would one know when to maybe give up or move on, and when to push more and hope for the best? So this is closely related to what we have been talking at the end of your talk.
Yeah, I mean, I believed in myself at the end of the day. Like, companies succeed and fail based on the entrepreneurs running them. So if you will have questions. Like, you'd be crazy not to question things. There have been many times where I've had a month, 2 months, 3 months, my credit cards don't work, my debit cards don't work. These people tried to take over my company. Then I went to these people and they tried to take over my company. Just like, oh my God, like, I can't take this deal, I can't do this. But at the same time, I'm like, wow, like, I'm just gonna have to, you know, I'm not gonna be okay for a little while here. And that's okay. Like, you have to be willing to decide, are you willing to make that sacrifice or not? If it's an app for, you know, I don't know, like delivering dog food or something, and you go through this stuff, you might be like, that's fine. Like, I'm just gonna move on. I got experience. Awesome, right? And there were many days where I'd wake up and I'd be like, if this were anything else, like, yeah, I would give up. Like, no question about it. If it were some kind of software company that doesn't have intellectual property or otherwise, like, yeah, I don't need this. I can bury it and start over. But if you're building something that has, like I said, a lot of intellectual property or long-term like R&D and development around it, that's years of work, you can't expect other people to always appreciate it. You can't expect investors to understand it. You just have to have, like, you know, self-esteem and know your own value and the value of what you built, and stick to it. And there's no other way to put it. You go for a walk, you know, go for a run, take care of yourself, like, practice good nutrition, right? Like, those are the things that actually you learn to do because you need to do that to survive. So you have to believe in yourself.
Thank you. So we have more one question. So the company has proved that the COVID, that this peptide is its efficacy in the computer modeling and simulation studies. So how far are you from? Yes.
Also.
Yes.
And
how far are you from validate this in the preclinical clinical efficacy of this technology?
Yeah, great question. So we, like I said, we also validated that the peptides can actually block an infection. So not just in silico and through the computational stuff, but we actually put live lentivirus onto the cells, right? Right now, we actually, I think I can talk about this. We have a contract with the NIH, so we'll be sending them materials in the next week or so, and we'll be getting data with, you know, BSL-3 live virus infection in next month. And then furthermore, we're also in queue with the NIH for performing these humanized ACE2 mouse studies, which then, after we have that data, the goal will be to go for an emergency investigative new drug application. So you go to like some severe or critical or maybe even moderate COVID patients, find a physician to file an emergency IND and try to basically put this directly into humans, even if it's a small trial, 3 people, 5 people, just getting it into humans and then rolling that into phase 1. So we're trying to do all of that by Q1 of next year, if not, you know, by the end of this year.
Thank you, Andrew. So
Thank
we do have a few more questions, but we'll post them for the panel discussion.
All right.
But I will now ask all the speakers to please turn on your camera. Okay, thank you very much for all the questions and that the audience make to the speakers, and as well as to the speakers itself for their talks, where they were really clear and engaging. So now we have reached the panel discussion, and in the next 20 minutes, more or less, we'll be just making a few questions about the entrepreneurship process itself, so that you can share your own thoughts on this. So to start with, I would maybe ask Tammy to first give some comments on this first question. So when did you start to think about starting your own company? What motivated you to start a company?
Yeah, I think when I joined grad school and was kind of looking around at labs and picking a project, I think it was always kind of a project that would have industrial relevance and could potentially turn into a company. I think I wasn't necessarily thinking so early that I would start a company, but I think as the project kept going well and it seemed really promising and seemed like it could have some really good real world impact, I think that's kind of when my thinking started to change. And then near the end of grad school is when I started doing things like applying for IndieBio, applying for SBIRs, and kind of going and looking at resources on campus like that.
Javier, can you please add from your side? We cannot hear you, Javier. No.
Okay, thank you. Yes. Thank you, yeah. Yeah, I remember it very well. I was sharing the hood, and the student who I was sharing the hood with told me that he will take some of his polymers to a hospital to treat some people with diabetes. And then I realized, wow, this is what science is really about. You know, I was fascinated by the fact that something that you are making in the hood could solve a problem, you know, that very day. You know, it was an eye-opening experience. And then I said to myself, okay, I remember you remember my presentation, right? So there is a catalyst, and I do a post-treatment, and that catalyst solves a problem, right? Basically, that's what we are doing. So that day, I decided to send 2 catalysts, my catalyst, and the one before being modified to a national lab. I'm getting echo now. Can you hear me well? Okay, yes. Better now. So I decided to send 2 catalysts, the conventional one and my catalyst, to a national lab in Canada. And that day I said, okay, if I get better results, I will start a company. And I got the results back. They were very impressive, and that was it. It was as simple as that. I was sharing a hood with somebody who showed me very clearly that what he was doing there had an impact, and I decided to do the same.
Thank you, Javier. Andrew, you want to add from your side? Can you unmute, Andrew? Cannot hear you. Yes.
Hi. Yeah, so I think starting a company, I knew that I wanted to start some kind of company or have some kind of business when I was like in school, right? Before I knew that I wanted to do this specifically. So there was definitely an idea of I want to be able to commercialize something, but at the same time, I don't want to just sell some product. Like money is not so important to me. Like if I'm gonna make money, it would be to do more of the thing that I want to be doing. Because really, there was very early on a couple realizations. One, you know, for quality of life, you could basically, you know, retire and live forever off $200K a year if you make like $5 million in the US. So you can live like a great life. You don't have to worry about anything. You can go hang out on a beach somewhere, right? Just also decided that's not really for me. Like I would get really bored really fast. So, you know, if I'm going to make money, I need to make a lot of money. And the reason I need to make a lot of money isn't for myself, it's so that I can like actually build impactful things that the world needs that most people just wouldn't try to build. So I think around April of 2013, I wrote my first business plan for Lycandle. That was like right before I finished my undergrad. And that was right when my research started to work also. Like the first, that was within a couple weeks of showing, you know, yes, these nanoparticles can be used to express a gene in a cell. Okay, like that is very exciting. Now let me spend the summer improving on that research, continuing on. And the goal is like, I'm going to start a company. And if all goes, it doesn't really matter. I'm going to just do everything I can until my one-way flight on August 12th of 2013. And then I'm starting a company. Like I have $1,000. I'm buying a one-way ticket to Silicon Valley. I'm going to figure this out. I don't have another option. Like I'll sleep on a couch or whatever, doesn't matter. So that's one thing. The other thing was that I was looking at statistics for professors, and like in the US, the average professor receives their first R01 grant, first multi-million-dollar, you know, multi-year grant at the age of 38. So I'm like, all right, so I'm 21. So I have like 17 years to spend at least $1 million a year on research, and I would be able to do more than, you know, like the average professor at 38. So I took that bet that I could accomplish that in industry, and here I am.
Okay, thank you all for sharing your thoughts and insights on this first question. So the next question is related to the following. So can you please point out the major challenges while building your company? Like what risks and sacrifices does such an enterprise demand? Maybe you can start with Javier, please.
So many challenges. For me, the most difficult one is, of course, the team. The team is a main challenge. I regret I didn't fire some people sooner. That is a very important decision. I'm very happy with my investors. The investors were very supportive, and my clients. But yeah, I guess that's the main concern and the main mistake. And then from the personal point of view, I was able to do both academia and entrepreneurship at the same time. So I was a faculty member here in Spain as I was having my company. So it was a very satisfactory experience. So I enjoyed a lot. I was able to see the discoveries made in the lab, and then talking to customers and that who wanted those products, right? So from my own personal point of view, it has been a wonderful journey. My main regret, I knew, and people in the company knew, we had some very bad employees, lazy people, not committed, and we didn't fire them. Of course, when my company was acquired, those were not hired by the next company, but we didn't fire them, and that was a mistake.
Thank you, Javier. Tammy, you want to add? We cannot hear you.
Yeah.
Yes.
Yeah, I would say for us, fundraising was not a walk in the park, I guess. That was definitely one of the challenges, but I think it so far has worked out. And yeah, I guess I would also add on that yeah, the team is one of the greatest parts, but also one of the most challenging parts. I think since I basically came right out of grad school, I think grad school is kind of a more solitary activity, and your project is kind of your own. And in a company, it's just like a lot of people working together and kind of bringing different perspectives and having different kinds of ways of doing things. And so having everyone work together and be able to kind of operate smoothly as a larger machine has been something that we've put a lot of effort into building out.
Thank you, Tammy. Andrew?
Yeah, definitely people, like hands down people, right? I mean, people make or break everything. Having one person who is there for the credit or kind of like hand wavy or yeah, I don't know, or like you've got your pontificator types who want to like argue every fine point and get a bunch of people around, and you know, you'll be like, hey, so today we want to do this and this and this, and this week we're going to accomplish this, and then you'll get you know, the oh, well, I think this should be broken down like this, and we should spend 2 weeks on optimizing this before we go after this. And you're like, it's not academia. Like, we don't have time to spend 3 weeks optimizing microscope for like a cell line. You know, like we need to move. Like, and there were definitely like a lot of moments of just, okay, like I don't care if I have 7 PhD scientists working with me from Harvard, MIT, and Stanford or whatever. Like, okay, like I'm gonna go spend an all-nighter in a lab and just get this data because no one else is willing to do it. So things like that. People, right? Fundraising, guess what? People. So now you're dealing with not just like people in your company, which can slow you down, but also you're dealing with now people who actively kind of have the opposite of your best interests at heart. So they can spend 3 months, 6 months, 8 months wasting your time. They can sign term sheets and change them after signing term sheets. They can get access to your financials and try to wait you out because they know how much money you're spending and when you're going to run out of money. So they go into like an exclusive deal with you and then they change the deal at the last minute. They can team up and try to figure out who you're talking to and then collude behind your back to try to take your offer and make it worse so that they can take control of your company. So basically you kind of go out with this believing in the best in people, and then you come out being like, wow, people are kind of awful. Like, you know, like it's capitalism in a way. It's not their fault that they're trying to make money, but they are, and they'll go to any lengths to try to make the most possible money in the shortest period of time. So, you know, you just have to go in knowing that it's like, it's not just like all this rosy world where you've got a great technology, people are going to love it. Like, there are more stories.
Thank you, Andrew. So we have here one question directly to Tammy, but I think that then we can extend as well to Javier and Andrew. So it's from Clybin Mari from Philippines. She's asking, were there specific challenges you faced shifting from academia to business? Would you suggest doing both at the same time or finishing studying first and then shift to doing business? Maybe you can start with Tammy.
Yeah, I think I did overlap for just a little bit, kind of transitioning from a grad school lab to a company. I think one of the biggest differences, well, a couple of biggest differences. One is kind of working in a team versus by yourself. One is basically the mindset of how is this going to turn into a product, and how can we commercialize it? How can we sell it? And how do we make it, I guess, usable and appealing to the market? What even is the market, and who are we selling to? These are the kinds of questions that it became a lot more important as a company rather than in the lab where it's like, is this experiment going to work? Can we actually make this molecule? So yeah, I guess kind of the mindset and as well as kind of the people aspect.
Andre, do you have something to add? Sorry, you are muted, I guess.
I was listening to Tammy, but the question cleared out from my head. Can you repeat the question, please?
If there were any kind of specific challenge you faced while shifting from academia to business,
right?
And if you would do it at the same time, or finishing studying and then moving to the business,
you're gonna learn more business in 3 months of trying to start a company than you would in like 2 years of getting an MBA, right? Like you'll cover the basics at like shallow depth if you go to, Harvard MBA or Stanford MBA or whatever. It's great. I mean, the main benefit to explicitly studying business and getting like an MBA is that you're going to have a network. So you're going to be able to email all your Harvard classmates and be like, hey guys, like, know anyone at this place? Or, you have your stamp, right? But in terms of actual like running a company, like you will learn running a company, especially a startup, which is not like I'm going to work in corporate land and, like you're going to learn everything that you need to learn very fast because you need to do it to survive. There's nothing teaches you faster. And that includes science, right? Like I have a degree in biomedical engineering. I formerly studied like biomaterials, worked in a gene delivery lab or, as an undergrad, but I learned more during my research in that environment, as well as after starting my company. Like I didn't have any background in immunology, but when we're like T-cell immunotherapy is what I need to focus on, like, yeah, you bet. Like I understand how T-cell receptors work and how like genomic recombination events work and how like the cells divide and how the affinity maturation works and how the engineering works and all the gene editing tools and like gene deletion, gene insertion, like all of that is mostly after starting a company. So like your education doesn't end when you finish your school and you get letters after your name. It's just beginning. Like you should be learning how to learn, not expecting, oh, well, now I finished school, I've learned everything here, and now let me go learn this other thing in like business school, and then I'll start a company and I'm gonna stop learning. Like, that's not how it works at all.
Thank you, Andre. So we just have 4 minutes until the end of this symposium. So I have just more 2 questions. So this one I would maybe direct to Javier. So it's from Rodal. What is the trait that you can say really help you to explore and work as a scientist entrepreneur?
So I didn't hear the first part of the question.
So what is the trait, the character that you can say that can really help to explore the business as a scientist entrepreneur?
Well, you need to be curious and resilient. I think those are the 2 more important qualities. But I also would like to comment a little bit about the previous question. I said I wrote that article for Science, how to become an academic entrepreneur. And I want to say that being an entrepreneur made me a better scientist and a better professor. So my students get a different kind of information, skills. I shared a different kind of passion for chemistry because I actually started a company, talked to customers, and students can feel and appreciate that kind of background that many chemistry professors don't have. Same thing now in my new role as president-elect of IUPAC, right? I sold my company, 10 years experience, as Andrew was saying. You get a kind of experience that no MBA is going to give you. So I was wearing these 2 hats, an academic hat, many papers, many recognitions. I've been teaching inorganic chemistry for 15 years now, but at the same time, I talk to customers and doing consulting job. I mean, you are just a more complete chemist. You are more round, you have more experience. And when you need to deal with customers, you don't have all the freedom that you have in the lab. And Tammy gave us a perfect example, a very nice chemistry, right? You can do things in the lab which are very toxic, any quantity, whatever you want to do, it is fine because you are going to produce only 1-2 grams. So whatever waste you produce, it can be recycled or deal with. But if you are producing thousands of tons, you need to make it cheap, and clean and all of that. So you have many more constraints. But that kind of training make me a better chemist, yes, a more complete chemist. So I don't know if I was a good chemist or what kind of characteristics I have before becoming an entrepreneur, but what I know is that these 10 years of experience, as Andre was saying, made me just a better scientist, a better professor, and a more round, more complete chemist. So now when I talk to my colleagues all around the world through IUPAC, I feel I'm talking to my colleagues, both when I'm talking to academia, but also to industry. And I'm invited to this kind of webinars precisely because of that, because I decided to do something different. That's why I insisted during my presentation about being okay to be different. Actually, I think diversity is very important in chemistry, and we need to change education. We keep teaching chemistry in the same way it was taught 100 years ago, one professor giving a seminar to hundreds of students. I think that needs to change. And why I feel that? Because I experienced that when I was going through my tenure. My university, the committee who was hiring me challenged me, what is going to be your commitment? You also have your company. Are you going to devote enough time to teaching? Yes, I will devote more kind of teaching. And guess what? My teaching is going to be better because I have this experience, because I know what it means to scale up. I know what it means not producing waste and circular economy, not because I read a book, because I actually lived it through. So yeah, I fully agree with Andrew and Tammy. I think they're absolutely right about these things. Yeah.
Thank you very much. So the last question, and I ask you to be concise, just like 20 seconds for each. So any last advice for all those that are looking for raising their own business? Maybe you can start with Tammy, please.
I think it's been a really great journey, and I would highly recommend it if you think that you have a good idea and you want to pursue it. So go for it.
Andrew?
Do it while you're in your 20s, right? Like, do it before you have to worry about, well-being of your family, or like, I'm married or got to pay like a mortgage on my house. Do it while you can take risks, because like the worst that happens is you don't succeed at building a company, but you gain a ton of experience. The debt and money that you spend isn't money that you personally owe. So, don't do it just for the sake of doing it. Do it to solve an actual problem, because otherwise you're just not going to make it. But like, if you're going to do it, then do it sooner rather than later.
I would say two things in 20 seconds. First, we need entrepreneurs. We need people that bridge the gap between discoveries and the market. We need them, right? And it's a fantastic journey. It's going to make you a more resilient, more knowledgeable person. But don't do it if you are not 100% convinced, because it's not easy, it's not rosy. It's a very difficult experience. So you need to be prepared for that. So I hate when people say, yes, go for the ride, try it out. You need to be convinced, because you're going to be asking for a lot of money. You're going to be asking people to work with you. So, and don't lie. Very important. Under promise, over deliver. Don't oversell your technology. Don't lie at any level. Just be humble, say what it is, don't oversell, once again, under promise, over deliver. Very important in this business.
Thank you very much to all of you. So we do have a few more questions to ask, but due to sake of time, we have to move on. So I just wanted to finalize this symposium by thanking very much all the speakers, Javier, Tammy, Andrew. Thank you very much for joining us and for sharing your expertise and the experience while raising your own business. Then I would like also to thank all the attendees for coming and for making questions within the chat. So to learn more about what you are doing at IYCN and YCC, please check the website we have here, so IYCNglobal.com and ACSYCC.org. You can always email us, so we'll be happy to hear from you. and please be attentive to our social media accounts, because we'll be running very soon some more interesting webinars that I'm sure that you don't want to miss. So thank you very much, and have a nice week.
Thank you. Take care.